Hello, and a very warm welcome to today's Select Science webinar titled Why Functional Assays Are Crucial for Biopharmaceutical Development Success. My name's Charlie Carter, and I'll be moderating this presentation. And I'm delighted to be joined by Therese Siegerstein and Ali Karma of Spar Life Science, who have a fantastic talk lined up for us today. Please submit your questions for the Q&A session at any time during the webinar using the button to the left of your screen. And without further delay, I'd like to hand over to Therese and Ali.
Hello, everyone, and welcome to this webinar titled Why Functional Assays Are Crucial for the Success in Biopharmaceutical Development. My name is Therese Siegerstein, and I work as product manager for our cell-based solution at Spar Life Science. And I have my colleague with me today.
I'm Ali, and I also am a product manager at Spar with the responsibility for the complement and calprotectin portfolio of the company.
We hope that you will enjoy today's webinar. If you have any questions, please use the chat box and we will try to answer them accordingly. Hope you enjoy it. So Ali, take it away.
So, as we speak, we are actually witnessing a paradigm shift in how we approach treatment of various conditions. There is an increased focus on personalized treatment and personalized medication, and with that comes the search for drugs that fit into this approach, much since we are on the lookout for more effective treatments while reducing the negative side effects of drugs, but also because we are reaching a peak in how we can use small molecules to combat novel diseases and conditions. This search has taken us down the path of an entirely new field in medicine, the biopharmaceutical drugs. Now, often mimicking biologically occurring megastructures, although there are some smaller biomolecules, these drugs are often safer and more specific than their chemically produced counterparts. And as these molecules rely on modifying biological functions, functional assessment and functional assays are required to show that a biologic fits the desired functionality and quality profile. So in this webinar, we will delve into this exciting topic and also look into how functional assays are needed for safe and compliant drug development, especially when it comes to biologics.
So what is biopharma? Well, the term biopharmaceutical was coined in the 1980s and refers to drugs that are produced in biotechnological processing using live cells and organisms, unlike the conventional drugs that are a product of chemical reactions and referred to as synthetic drugs. These are complex medicines and can be proteins, including antibodies, nucleic acids such as DNA, RNA, or oligonucleotides, that are used to prevent, diagnose, and treat various conditions. The most common type of biopharmaceuticals are monoclonal antibodies, and this is also the highest selling class of these drugs, with currently more than 1,500 drugs in the product pipeline. Other common types of biopharmaceuticals are recombinant version of human endogenous proteins and hormones. A recombinant human insulin was actually the first biopharmaceutical approved for human therapeutic use in the 1982. In recent years, a new class of biopharmaceuticals operating within cell and gene therapy has increased. Different therapies such as CAR-Ts and AAV vector-based therapies has gained a lot of clinical success and interest from the pharma industry. As mentioned, today the biopharmaceutical industry is enjoying considerable success. Its products account for about a fifth of the world's pharmaceutical revenues, which are growing at twice the pace of those generated by most traditionally chemically synthesized drugs. These type of drugs populate the list of best-selling drugs and a number of these have achieved blockbuster status. So why the success story? Well, there are many benefits to biopharmaceuticals, and we will try to explain what they are in a bit more depth in this webinar. But most importantly, it's the ability of these drugs to be specific to their targets, to mimic normally occurring structures in the human body, and in many cases make use of the body's own cleverly designed defense and protection system that makes them so useful.
Small molecule drugs and biologics differ substantially in just about every single aspect of their attributes. Now, fundamentally, they have different physiochemical properties, which not only affects the mechanism of action, pharmacodynamics, pharmacokinetics as well, but also affects the safety and efficacy of the drug, and even impacts product manufacturing and quality considerations. It also affects the handling, storage, and shipment of the final product, as these biomolecules, similar to the biological structures they are mimicking, are sensitive to temperature changes, prone to degradation, aggregation, and so on. And seeing that these drugs are produced in live host cells, there's also a potential of batch variation, causing a lot more stricter rule sets during approval and distribution. Now, the pharmacological activity, stability, and permeability of small molecules largely depend on the chemistry of the molecule as opposed to the structural interactions of the biologics. For the biological compound, the pharmacological activity, stability, and permeability is influenced by the molecule's primary through to tertiary structure. So amino acid sequence, the local twists and turns, the alpha helices and beta sheets, and then the overall 3D structure and the folding of the protein or the molecule where that is applicable. So these differences are quite significant and quite apparent. Now, due to this, there is one benefit with conventional drugs, which is really important for quite a few, and that is their bioavailability. Due to their properties, the conventional drugs can be administered in quite a few ways, and it is also possible to do oral administration. For biologics, this is not possible. One, because the molecule is quite big, so it won't have the capacity to get through into the bloodstream if orally administered. And the second one is that its physical properties will not allow it to do so. So biological drugs hence need to be either administered through intravenous injections or as subcutaneous medication. Now, the latter is something that diabetic patients have been doing for ages, and it also allows for self-administration, just as diabetic people have done with insulin shots. Still, there is a barrier that needs to be crossed for the patient, with either having to go into the hospital or the clinic to get a shot, or to poke themselves with a needle. But apart from the needle bit, the biological drugs have some major benefits, and this is what that have been contributed immensely to the success of these drugs, and that is the ability to target conditions that previously were not possible to target by conventional drugs. The personalization of treatment, and the fact that these molecules interact with the normal existing pathways in our body as part of the pathways and not as foreign objects or foreign molecules, and hence reduce the number of side effects caused by the treatment. So, we will come back to the mechanism of action for biological drugs in a bit, but I would just like to add in here, since I mentioned it in this segment, that biological drugs are produced using a wide variety of live cells. So it might be bacteria, yeast, mammalian cells, even insects have been experimented with. And through this, scientists and pharma companies have managed to produce drugs that firstly can introduce biologically active molecules into patients who are missing them, such as in case of insulin into patients with diabetes type 1. But they can also use this methodology to create immunosuppressive drugs to calm down an overactive immune system, which is the case in chronic inflammation such as rheumatoid arthritis and IBD, inflammatory bowel disease. But they can also use the same methodology to help our immune system find, identify, and kill cells that are damaging to us, such as cancer cells. So this approach to personalized medicine is really does really have huge potentials. And as time goes by and there is development in methodology and also in reducing the cost of producing it, we will see more and more drugs popping up based on this approach.
There is often talk about biologics and biopharmaceutical versus small molecules, referring mostly to synthesized drugs. But even within the biopharmaceutical group, there are many different categories. In the wake of the success of biopharmaceuticals in general and monoclonal antibodies as the therapeutic agent in particular, a new group of therapeutics called biosimilar, also known as follow-on biologics, has emerged. A biosimilar is a biological medical product that is almost identical to a copy of an original product but manufactured by a different pharmaceutical company. It is highly similar to a licensed reference product, which is called the generic drug, and this generic product is usually the first on the market and holds the patent for this application or MOA. The biosimilar may show minor differences in clinical inactive components, but the key is that there are no clinical significant differences between the biosimilar and the reference products in terms of the important parameters such as safety, purity, and potency. And showing this biosimilarity is an important part of the analytical package for any drug company seeking to register its drug as a biosimilar. The first biosimilar, somatotropin, a growth hormone, was registered in the EU in 2006, and the first monoclonal antibody biosimilar, the drug infliximab, which is a medication used to treat a number of autoimmune diseases, was registered in the EU in 2013. There are today approximately around 400 approved biopharmaceuticals on the market and around 50 approved biosimilars. As companies see a lot of potential revenue that could be generated from biosimilars to current blockbuster drugs, there is an extensive pipeline of biosimilars coming out in the next couple of years to blockbuster drugs that run out on their patent. As an inevitable twist when developing these biosimilars, sometimes improvements compared to the original was made. This has given rise to the term biobetters, which refers to therapeutics from the same class as an existing biopharmaceutical, but it's superior to the original. Hence, it's neither an exclusively new drug nor a generic or biosimilar version of that drug. Instead, you'll end up with a drug that is intended for the same therapeutic area, but with added benefits. What is MOA and what is an MOA refractive assay? In order to properly evaluate and understand the clinical effects of a biopharmaceutical, the drug's mechanism of action is paramount. Hence, it is important to know that it works, but also how it elicits its therapeutic effect. What is the mechanism of action, MOA, of this drug? The growing consensus is that the selection of an analytical method should be driven by a product's intended physiological or therapeutical mechanism of action. And in cases where the MOA is a simple binding to a target, a surrogate method such as a protein binding or competitive binding assay may be very well sufficient. But in cases where a more stepwise reaction is happening for the drug to induce its effect, an assay that more closely reflects and mimics these steps is needed to properly reflect the biological context and the physiological reactions. This is referred to as an MOA reflective assay. Selection of an appropriate MOA reflective assay method to determine and assess these complex biopharmaceutical has its challenges. Not only do you need to mimic an often complex and sometimes not completely understood mechanism of action, but also because these bioassays can be costly to develop, difficult to perform and transfer, and to maintain. And unlike other analytical techniques, bioassays are almost always unique for each therapeutic compound. On the other hand, a well-designed bioassay will accurately capture the biological activity of a drug candidate and give you more value for your sample or value for your measurements, since it allows you to actually follow and quantify a series of events instead of just a momentarily snapshot. It's like imagine being able to monitor the falling domino bricks in a row instead of just seeing the moment where sort of the first push is being made. A key thing in understanding what mechanism of action that is required in an assay during drug development process and post-launch release, we first need to know how various biopharmaceutical works, and Ollie will take us through that.
The mechanism of action of biological drugs obviously depend on what the drug is intended to do. As in case of insulin for diabetic patients, the mechanism of action is just that, to supply insulin. But in some cases, especially when using antibodies, there will be differences in mechanism of action, and there will be different approaches depending on the target and where it is and so on. So I will just give you a few examples of existing drugs and how they work, just to show you the broadness of application. The first one I want to start off with is a drug called benralizumab, which binds interleukin-5 and is commonly used for patients with asthma who do not respond to standard treatment. And the reason for this is that it is desired to prevent interleukin-5 to bind to eosinophils and have them activated. So in this mechanism of action, the ligand-receptor interaction is blocked by a ligand-binding antibody. Similarly, we have tocilizumab, which is a different example of disrupted signaling between ligand and receptor binding. But instead of binding the ligand, this drug or this antibody actually occupies the interleukin-6 receptor, so it is or interleukin-6 antagonist. Interleukin-6 is a pro-inflammatory cytokine, and this drug is hence commonly used in various chronic inflammatory conditions, but also in other indications as well, where interleukin-6 is a key player in the disease activity. A completely different approach is taken by rituximab, which induces antibody-dependent cell cytotoxicity, or ADCC, and complement-dependent cytotoxicity, CDC, through complement classical pathway activation. And this is achieved by binding to the CD20 portion of CD20 plus or CD20 positive B cells in order to remove malignant B cells from circulation. Now, all mature B cells have CD20 expressed, so it will also remove the healthy cells, but these will be replaced by next generation B cells, be healthy. Fourth example I would just like to present quickly is trastuzumab, which acts similarly to the previous example, but slightly different. So trastuzumab induces antibody-dependent cell phagocytosis, ADCP. And this one is commonly used for in breast cancers, as it binds to HER2, which is expressed on cell surfaces in certain breast cancers. So when it binds, it will bind to the malignant cell, and then it will also bind to a phagocytic cell on the other end and facilitate phagocytosis. So as you can see in these examples, the antibodies can be used quite diversely depending on their target and their intended use. But what we also see is what is implied when we talk about personalized medication or treatment, because none of these drugs would have been given to a patient who had not been confirmed to have the target biomolecules, either in circulations as interleukins and other cytokines and so on, or cell surface expressed molecules, as in the two latter examples. Now, depending on what the target is, the mechanism of action would change accordingly, but either way, it would be a part of what would be a natural process in the human body, just that the target would not be natural in the sense, because the body was not able to produce antibodies against those targets, so we had to introduce them for that. So it does give an idea of how versatile biologics are. These examples also show one more thing, and that is the importance of having functional assessment of drugs, especially biologics, with correct mechanism of action, their real-life target, so to say, in order to ensure that the drugs actually work the way they are supposed to be. It's not sufficient to just have a quantification, concentration, or identification of the drug in and of itself that is there, but it actually has to be a functional assessment. But this, I believe my colleague Therese will go a little more in depth into, so I will leave that with her.
The need for functional assessment has become very clear with the development of biopharmaceuticals. As previously mentioned, these drugs can operate on many different mechanisms, but often target endogenous pathways, either by enhancing or suppressing a given signaling molecule. This requires an assay that can show a stepwise reaction, and also that the assay is performed with the proper relevant biological background, so all the intracellular pathway activity can be assessed and investigated. This need for biological relevance per se, it's not something new, and there are many approaches to try to accommodate this. The use of primary cells, which are a direct isolation of cells from a donor, will get you the most biological relevant setting and is widely used. The benefits of using a primary cell assay is that the primary cells maintain many of the important markers and function we see in vivo, and they are less prone to mutations due to fewer passages and a shorter lifespan. However, primary cells have notoriously inherent variability, and the efficacy varies greatly between different cell types and cell donors. In addition, they are difficult to grow, as well as having a limited expansion capacity. In recent years, innovative assay formats such as reporter gene and second messenger assay has emerged as drug discovery tool. What is characterizing for these assays is their ability to measure the regulation of cellular signaling events upon drug treatment, instead of just analyzing the respective downstream outputs such as proliferation, toxicity, or cytokine release. Commonly used cell-based reporter assays are based on stably transfected cells using vectors encoding for luciferase or other types of reporter gene Gene expression is then controlled by an inducible promoter, where the production of reporter protein is directly related to the binding of these transcription factors involved in the MOA to the corresponding response element. The output of these assays are easily quantified by common plate readers, and the simplicity combined with shorter assay times, we are talking hours here instead of days, and improved reliability and high sensitivity, as well as larger dynamic ranges, are some of the key advantages of these assay format. With that said, it's important to stress that while reporter gene and primary cell-based assays are the most common assay within this field of MOA reflective assays, ELISAs can be used for functional and biological assessment as well. And there is no need for a contradiction position between these two types of assays. Rather, they will both provide part of the whole information package needed. For example, a ligand binding or a quantitative assay will indicate whether or not a molecule is present, and a functional assay will show whether that molecule is actually functioning as it's supposed to. It's a brilliant teamwork.
Yes, so as Therese mentions, it is definitely a brilliant teamwork between the functional and the quantitative assays. But let us discuss these functional assays for a little bit. So normally, ELISA is a quantitative assay, and it is a well-established method for this purpose. What it does not do is to give you a functional readout. So you do know something is binding, but you do not know if that something that is binding does actually have the activity that you want it to have. Now, as an exception to this rule, is our functional assays for complement activation, where you can do exactly that, get a functional readout. The basic principle of these assays are that we can activate every single pathway of the complement system individually and measure the end product in terms of TCC. Then, when drugs are added into the solution, we can see what kind of effect the drugs will have on the complement system. Will it have, if it is supposed to or intended to have an inhibitory effect, we will see a decrease in one of the pathways towards the end product of it. And this effect doesn't have to be directly, it can also be indirectly as well. But there are ways to go around to do this. But for this reason, we can then use the ELISA setup to get a functional readout. But as you might understand, it is always it will still be limited to the complement system, and obviously drug target interactions are way beyond that. So a more generalized approach where you can test for just about any drug target is by using cell-based assays. These are a bit more complex and have a little bit more complex approach, but the result is highly specific and it very well mimics the reactions that you can expect in the human body. So does the drug actually work or not? And is there any cross-reactivity and so on? So the principle for all and any cell-based assays are quite similar. It also includes our iLites, which you can see from this presentation. What you can see is that what we do is that we will have a target-specific receptor gene transfected into the host cell, along with a reporter gene construct. In our case, we use firefly luciferase, and in some cases, you can also have a secondary report gene called normalization gene. In this case, it's Ranella luciferase, which is also included to reduce the matrix effect, as well as to give a stable background for the solution. The target-specific receptors are expressed on the cell surface, and once our target binds, it triggers a cascade of signaling reactions within the cell, ending up in expression of our reporter gene, so firefly luciferase. And this expression can then be measured using a substrate and an illumiноmeter. Now, this will result. This setup will result in an assay that gives a functional readout as an end signal, but this could not occur unless there is a drug-target interaction. And because it is dependent on this, the sensitivity and specificity of this assay is really, really high. One of the other great benefits with this the cell-based assay approach is that the ability and possibility to customize it to fit just about any kind of need that would arise or any functional assessment that would be needed in a drug development process.
Why and when is a MOA reflective potency assay important? Guidelines define potency as the quantitative measurements of biological activity which is linked to the relevant biological properties, and that an assay demonstrating this biological activity should be based on the intended biological effect, which in turn should be ideally related to the clinical response. Oof, that's a lot. Well, the general interpretation is that a drug's potency will reflect how well or strongly, usually in relation to a set standard, a drug can perform its desired effect. And this might be a bit simplistic, but for the sake of understanding, let's stay with that. Furthermore, it is stated that a potency test shall consist of either an in vitro or in vivo test or both, which have been specifically designed for the method and should be specific to the mechanism of action of that biological being studied. Measurements of potency plays an essential role in the whole development chain, from early development phase into clinical and subsequent quality control, GMP product release testing, and stability testing. Biologically, activity or potency is a critical quality attribute that often faces increased scrutinizing by regulators. And to meet these requirements of regulatory submission, the potency assay should be validated to set guidelines and international standards. There are many types of assay that can fit the MOA reflective description: cell migration assays and cell signaling assays, proliferation and inhibition assays, binding or competitive assays, and ligand and receptor binding assays. Using an example from our own reporter-gene assay, our iLite cells, as they are a cell signaling assay, we can here illustrate an MOA reflective potency assay. So, when stimulating the cells with the drug target of choice, an increased luciferase signal will be obtained with a higher concentration and a sigmoidal dose response curve generated. This can be used as a reference or be compared to a set reference to assess the relative potency of this compound. Hence, how far off is this particular batch or composition from a set reference standard? Consider a more straightforward approach is using ELISA assays. They can also be MOA reflective and will allow for direct measurements of the biologic's affinity to its target with great accuracy. It's not uncommon that more than one type of potency assays is needed in order to properly reflect the drug's mechanism of action and to fully understand how the effect is achieved and subsequently quantified and standardized.
So we need to keep in mind our first line of defense when it comes to fighting off foreign substances: the innate immunity, and more specifically, the complement system. Any foreign substances that are introduced will have the potential to trigger this system, and biologics are by no means any different. Unwanted reactions to drugs can occur for various reasons. One of it is by it doing what it is meant to do. An example of this is rituximab, for instance, which we have discussed earlier, have been shown to potentially give moderate to severe side effects if the number of CD20 positive cells are present in high number, because that will lead to the activation and overactivation of the complement system. That needs to be considered are issues with impurities in the final products. Pharma companies obviously will do their utmost to prevent these from finding the way to their end users, but this still might occur. I mean, there are, of course, guidelines for how to prevent this from happening and what is acceptable, but the impurities that do find their way can trigger an immune response. So the impurities in drugs, just to mention, that might be from different sources. Now, it might be different molecular variants of the product that is being produced, or the drug that is being made, cleavage products or truncations and so on. It might be aggregates, or even it might be degradation products from any stages in the manufacturing process, all the way from start up until the point of injection. So it really is impossible to prevent some sort of impurities to find their way to the patient. But these impurities can also include host cell molecules, such as host DNA or fragments, or equally culture molecules, cell culture molecules, such as inducers or media components. Point is, there are a lot of possibilities for impurities to get administered into the patient, and hence it is really important to keep an eye on the complement system to see how it reacts to the drug, even if it's not intended to be a target for that drug at all, but just to see if it is activated without that being the mechanism of action for the drug or not, or in the other end, if it is inactivated in some way or inhibited in some way, when that is not the mode of action for that drug. So the complement system is really important, and it should be kept in the loop in all stages of development, and even after once the drug is marketed, for each and every batch release to test for the mentioned issues.
Another major application area where functional assays are key is within immunogenicity. There are many factors that influence the immunogenicity of biopharmaceuticals, and although in most cases an immune response to a biopharmaceutical has little or no clinical impact, they do pose several potential risks for the patient, particularly in the case of anti-drug antibody and neutralizing antibodies. This immunogenetic response, where antibodies are produced by the immune system as a protective reaction, can adversely affect the pharmacokinetics, bioavailability, and efficacy of the drug, and may neutralize the activity of the drug. Assessment of drug immunogenicity is a regulatory requirement and is considered part of the safety evaluation that all biopharmaceuticals drugs undergo. The standard approach used to measure ADAs and NAb is in a multi-tiered approach. So during the first tier, a screening assay is used where focus is on detecting anti-drugs antibodies, ADA, that bind to the biological drugs. This is usually carried out in a ligand binding ELISA assays to be able to detect all types of binding. Positive samples from this initial screening indicating presence of ADAs are then rerun, usually on the same platform as a confirmatory assay. The confirmed ADA positive samples then need to be further characterized in order to understand what type of binding they have and to predict and evaluate any clinical significance this might have. Here, a functional assay, usually a cell-based assay, is needed, since you need to be able to see if the antibodies present in the sample will have any effect, and if so, to what magnitude. Hence, will it prevent the drug from binding or being active, and in worst case, may it cause serious adverse effects. Again, using our iLite cell as an example, we can here illustrate a neutralizing antibody assay. So, similar to the potency assay, the signal will be generated once the cells are stimulated with their target of choice. But if there are neutralizing antibodies present in the sample, the amount of free drug to stimulate the cells will be reduced. And this will result in the quenching of the signal and a decline in the curve with increasing amount of neutralizing antibodies present. By combining a ligand binding assay and a cell-based assay, a full characterization of ADAs and NAbs in the requested tiered approach is achieved. Biopharmaceuticals represent some of the best accomplishments in modern science. These drugs are increasingly being used in practically all branches of medicine and have become one of the most effective clinical treatment modalities for a broad range of diseases, including cancer and metabolic diseases. And what we have tried to highlight here today is that developing a bioassay that can be used to fully understand how these compounds work and induce their effect is a complex, rigorous procedure that needs to address many challenges.
So one of the first one is to actually understand what we are building and to understand the multi-layer and complex structure of these megamolecules per se. So we touched upon this when we talked about the primary, secondary, and tertiary structure of the molecules, as well as the post-translational modifications of each molecule and how they, all of this combined, can affect the drug's functionality and efficiency.
Furthermore, in order to properly evaluate and understand the clinical effects of a biopharmaceutical, the drug's mechanism of action is paramount. Like we mentioned before, it is important to know that it works, but also how it elicits its therapeutic effect. What is the mechanism of action of this drug? And the growing consensus is that the selection of the analytical method should be driven by the product's therapeutical mechanism of action, which means an assay that in the best way possible reflects and mimics biological context and physiological reactions.
As we discussed in one of our previous slides on mechanism of action, they can be quite different and broad in applications. Now, one of the easiest one, simplest one was ligand binding or interaction with soluble molecules such as ligands, enzymes, and cytokines, etc. But the drug could also well bind to the receptors to have an antagonistic and inhibiting effect. On the other hand, we looked at how these drugs can bind to target cells that the human body should get rid of in order to be preserved, such as cancer cells and so on. And this can be facilitated through different means and mechanisms of action, either by complement activation or phagocytosis. The point is that when developing a drug, the assays or the assays should include modeling all relevant mechanisms of action associated with that drug that is being developed in order to ensure correct functionality and efficacy.
Yes, and this becomes even more evident when looking into different specific applications and related guidelines. A potency assay should, by regulatory requirements, reflect the MOA of the drug and also give substantial information in order to support both early on decision as well as post-launch batch release and stability studies. And from a patient safety point of view, It is vital and a requirement to detect and investigate any clinical impact of ADAs and NAbs, since this might heavily affect the safety of the patient, ranging from a drop of efficacy to way more serious effects caused by an immune system going off spin. And then we have the issue of impurities that can cause adverse effects, which necessitates the need to monitor immune response and complement activity, even if it's not affected by the drug itself.
So there are many factors to consider when developing biologics from all different angles, but it's also important to keep in mind that this field is an exciting and fast developing area from both scientific, medical, and technological point of view, and as well from the patient point of view as well. So there are many new innovative solutions that are coming up, and as I also said earlier in one of our parts of the presentations, we're only sort of touching the tip of the iceberg on what we can do with these solutions, all with the common goal of getting a better, safer, and more efficient therapeutical approach that can benefit the patients who are in need.
So before we wrap up, a few words about Svar Life Science. We are a life science company that offers solutions in all of these suggested applications and has a long and solid track record of assisting pharma companies and other life science companies in their drug and product development, with a focus on biopharmaceuticals. The company group comprised of Svar Life Science, Wieslab, and Calpro, and together we offer a very versatile product and service offering. The core idea of the company is to be a partner and solution provider, ranging from off-the-shelf analytical assays and kits, such as our assay-ready cells for a Portogene assay and our functional complement kit, all the way to a full-service provider through our bioanalytical service department and customized cell and assay capabilities. We hope that you have enjoyed today's webinar and encourage you to reach out to either of us or visit our homepage if you have further questions or inquiries. Since Svar means answers in Swedish, if you have questions, we can help you find the right answers. Thank you.
Excellent. Thank you both. It's now time for the final part of today's session, which is our Q&A. And just before we start, we do have a question that's come in saying, will the slides and the recording be available afterwards? And yes, it will. The webinar will be available on demand in a few days' time. So please look out for an email from us in your inbox. And to kick off the Q&A, we have the question, what would you say is the key thing to take into consideration when choosing an assay for your analytical package?
Hi, my name is Michael Schwenker, Chief Technology Officer at Svar, and I will fill in for Therese since she is not doing so well and lost parts of her voice. I'm happy to take your questions. So coming to this very important first question, generally those assays tend to be very different and it needs to be fit for purpose. You always need to look at the composition and mode of action of the drug you are assessing and plan your work after that. In addition, it might be important which stage your assay will be used in. So as an example, preclinical animal work will have different requirements compared to neutralizing antibody assays in advanced clinical stages. However, the good news is we have extensive experience in supporting all types of bioanalytical assays at all these different stages, and our experts will be happy to support you through these decisions.
Thank you, Michael, and thank you for stepping in Therese's absence. Our next question is, what is the trade-off when it comes to biological relevance and ease of use?
So it's Michael again. As a general rule, the closer you get to a biological system, the more variance you are introducing and vice versa. So yes, it is always a trade-off and needs to be based on what data it is and what you want to achieve with this assay. Do I need to try to mimic what happens? Then a more biological assay should be used, or do I need a more reliable result to make a decision or comparison? Very often assay requirements are a question of getting acceptance via regulatory authorities. So again, we have developed a lot of these assays, and we try to consult and help in understanding what is needed for this particular assay. We commonly have extensive discussions up front, and we try to make sure to understand the assay and, what is often more important, the intended use of the outcoming data.
Wonderful. Thank you, Michael, again. We have an attendee here wondering, why have you put in a second reporter gene construct in your reporter gene cells?
An excellent question. Of course, what is very obvious is that the specific signal is driven by the firefly luciferase that is directly induced by the target we are assessing. However, very often people who have worked with these assays, they know that the matrix can take influence and it can affect the cells. So therefore, we have a secondary gene inserted, so-called normalization gene, and this allows to monitor for unspecific activation and other cell-impairing matrix effects. Another use is it can be used for troubleshooting. It will give you insights of the status and variability of cells. So for example, having this very low renilla signal might indicate that your cells have a very poor health and very poor viability. So it is addressing several challenges having these two reporter gene constructs in the cells.
Excellent. Thank you, Michael. Our next question is, do you have experience building potency assays for other types of therapeutics as well, such as bispecifics or AAV vector-based?
Yes, we actually have. So, of course, in the last 15, 20 years, many biologics were generated. Let's call them the classical biologics. But we are also working with, let's call them next generation biologics, biologics 2.0. So as an example, we are working with tri-specific antibodies. So one idea might be that you have a signal activation and a co-activation signal into an effector cell simultaneously. And we are using this 2-luciferase readout we have heard before to address both signals simultaneously, meaning the activation signal and the co-stimulatory signal. As a second example, we are working a lot in the area of gene therapy. Our reporter cell technology, iLite, is so versatile. We have developed several cell lines answering to transduced gene products, which are often used in gene therapy approaches. So this technology enables us to design a tailored solution for a potency assay in this promising new therapeutic area.
Lovely, thank you, Michael. Our next question here is, how are your functional complement assays different from other commercial assays that are targeting the complement components?
So, as mentioned so it's Ali here. So as mentioned in the presentation, the way our assays stand out is that they are functional in and of itself, and we are quite unique with that on the market, that we're the only ones supplying functional assays for all three pathways that you can run individually. And combining these three, you can actually narrow down what your drug will affect in the complement pathway to a few molecules. So by running all three, looking at different tables, what is activated, what is not, you can actually narrow it down quite well. What is also quite good with this assay is that although different in approach from standard ELISA or sandwich ELISA, it does not require much more effort than a standard ELISA. It's quite similar setup, just that the readout is either qualitative or semi-qualitative, and not necessarily a number that will give you a concentration. So I guess that's what puts us a little bit on a different level, that we have the possibility to sort of activate and investigate all three pathways separately and combine the results and output.
Wonderful. Thank you, Ali. Unfortunately, that is all we have time for today. So a huge thank you to Therese and to Ali for today's informative presentation. And thank you to Michael for joining us for the Q&A session as well. And thank you to everyone who's joining us online. I can see that there are still lots of questions coming in, which is great. And we will follow up with you with your answers after the webinar. Please also feel free to email me at editor@selectscience.net with any further questions. And if you'd like to share your attendance or provide feedback using social media, please feel free to mention us using @SelectScience or #SelectScienceWebinars. Remember, you can also download your certificate of attendance in the related resources tab. And if you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days' time. So goodbye and thank you all once again for joining.